So we're going to start talking about our live studies that we do in humans, which is almost exclusively done through a technique called brain imaging. So we're going to start off with computer tomography, or a CT scan. So this is one that you are probably fairly well familiar with because it's a pretty commonplace machine. Also, it's fairly often used in a lot of doctor dramas. So if you're a fan of Grey's Anatomy, you have definitely heard about this machine and the types of scans that it does. So a CT scan is going to be using X-rays to try and look at the different structures within the brain. So it's building up a picture of the brain using a bunch of different slices of X-rays using the differential absorption of X-rays. And what it means by differential absorption is that some parts of the brain will absorb X-rays more than other parts of the brain. And the differential absorption leads to either, so here in this image, that white ring around the outside would be our skull, and bone is going to absorb a lot of X-rays. And then we have darker spots here towards the center, and that would be very little absorption of X-rays. So that stands out from the lighter parts of the brain, which have sort of a moderate amount of absorption. So different structures have different compositions, and those different structures will absorb more or less X-rays that let us see the differences between those structures. Now, this isn't really good for finding very minute detail. It's only going to give us basically the gross features of the brain, and that means the overall general structures. But it's not going to give us lots of detail. It's not going to give us a lot of refinement sort of within those structures. And even sort of looking here, it's not even going to give us a really good definition between, say, the different parts. We'll use the terminology later, but you can kind of tell the hemispheres apart if you know what you're looking for, but you're not seeing a difference between, say, the lobes or anything like that. So basically, it's giving us an overall broad look. You'd see if there was something wrong, say a brain bleed or a dense mass of foreign material like a tumor. That would show up on one of these scans, but that's kind of it. Our next type of brain scan is going to be a positron emission tomography, or a PET scan. And so the PET scan is going to build up a picture of the brain using trace amounts of a short-lived radioactive material. And you don't have to panic when you see radioactive material. It's usually one of the very low-level radiation ones, and not giving off the kind of radiation that would be harmful and, say, cancer-inducing, but more radioactive in that it would show up to a machine that's designed to pick up excess energy, because that's what it's attuned to. And so we get this kind of fuzzy-looking color image over here. And PET scan is interesting in that it isn't just giving us sort of a stationary, here are the structures, it's actually giving us a primitive map of the functional processes going on in the brain. So the areas that are sort of this red color are more active, and then the blue color is less active. So we can see generally which area of the brain seems to be more or less active at any particular time. Now, the resolution on this isn't fantastic, so clearly our resolution of structures isn't great, but also our time resolution isn't perfect either. And with most techniques that give us an idea of function, there's going to be a little bit of a delay while we wait for blood flow and fluid movement within the brain to react to where we're active or not active. So this is usually a couple of seconds behind. So if you asked somebody a question, their brain wouldn't immediately light up on a PET scan. It would take a second or so to show up. So you just keep in mind that you don't have the best time resolution here. Our next one is another pretty common one. These are our structural magnetic resonance imaging, or MRIs. And so you'll see between this slide and the next slide are actually two types of MRI. We have our structural MRI, but we also have a functional MRI. And so the functional one adds a lowercase f in front of it so that we can tell them apart. Most of the time when we talk about an MRI, we're just getting the structural side of things. And so this is kind of like a CT scan because it's building up a picture of the structures, but this time, instead of using X-rays, we're actually using strong magnetic fields. So an MRI is going to take pictures of our brain and the structures using very strong magnetic fields that pass through the tissues of the brain. And so this is going to give us a much clearer view. So this is much nicer than what we were looking at with our CT scan. So it's going to give us the gross features of the brain. So it's going to give us the overall shape. We can actually see the two hemispheres here. We can see some of those middle structures, but this time the resolution here is much greater. So we can not only see the different parts, but also details within those parts. We can see all of the little nooks and crannies of the brain, and we could start differentiating different parts of the brain from each other. And we use this structural MRI. I almost called it an SMRI, which I suppose would also be correct. But this MRI is used sometimes in conjunction with the functional MRI as sort of a roadmap that you can base an idea of function on top of, but an MRI on its own is also still really useful. So if you had damage to one of the parts of your brain, you might be able to see it by looking at the overall structure of your brain. So this one isn't just used in conjunction with fMRI, but it can be used together, which leads us over to our fMRI. And so here you'd have sort of your structural MRI forming the background, all of the gray stuff that's giving us the map of the brain. That would be the structural MRI part of it. But the fMRI is giving us these red spots. And these red spots are showing us where our brain is active. And so for an fMRI, we're detecting changes in blood oxygenation and blood flow. So if we take a step back to break that down, we had talked about the fact that our brain is very resource hungry. And when we think hard about something, when we use a particular part of our brain, that part is firing more rapidly. The neurons are more active, and active cells need more energy. So they're going to draw more resources from the environment. So one of the major resources that we can look at is oxygen. And so if these areas are active, they need more oxygen to create and use energy. And so they're going to pull lots of oxygen in. And so these areas that light up red have drawn oxygen-rich blood to that area, and they're starting to remove the oxygen from that blood to be able to use it. So our high activity is going to show up as high oxygen use and high blood flow. So there's lots of blood moving in these areas because the cells in those areas are using lots of resources. And so we can see it that way. And a couple of other techniques that use functional detection are going to make some use of this trait where high activity in the brain means high oxygen use and high blood flow. And we'll actually see that right away in our next slide. So this type of imaging is one of the coolest ones because it's so different from all of the others. So this is diffusion tensor imaging, or DTI. And this is one that you don't see a whole lot of. But DTI builds a picture of water movement in the brain, and this makes use of an MRI scanner. So it's kind of working along the same idea as our fMRI, but this time, instead of just telling us which part of the brain is presently active, it's going to give us an idea of movement. And that's the cool part. So if you look over here, it's a little bit difficult to see until you know what it is you're looking at. But we have the general shape of the brain. And here we have what kind of look like, if you squint really hard, they're kind of like spider webs, where you have these strandy, sticky, outy bits towards the edges, and then they seem to connect inward and across the brain. So each of those would be a neural tract. And like we said in last lecture, a tract is just a nerve that's within the brain. And that's because of the weird nomenclature where they decided that when it's in the body, it's a nerve, and when it's in the head, it's a tract. But a tract is one of the bundles of neurons that's traveling together. And so each of these sort of stringy looking bits would be a tract or a bundle of neurons. And we're watching blood flow along those particular tracts. And so the different colors correspond to higher or lower rates of flow. But we can actually see in real time, so this is just a stationary image, but you could watch a video showing where blood is flowing. So you're not only seeing which area is high activity because it's drawing that blood, but you're also seeing where the blood is coming from. So you can see how it's moving through the brain. So you could use this to see where maybe blood flow is disrupted, or maybe where it's acting strangely. And so in addition to the really great part of being able to see movement of that blood flow, this type of imaging is also going to give us this high spatial resolution. And that high spatial resolution pretty much comes with anything that's tied to our MRIs. They tend to get really nice, clear resolution of all of our structures. But we also like this one for its directionality. And now we're going to start getting into some of the weird ones, because the first batch of them are, the standard. You think of them as part of a normal medical setup. You go into an MRI and you get your brain scanned in the giant machine and all of that. That's pretty standard. The next couple are different, and you'll see why as we talk about them. The first different one is near-infrared spectroscopy, or NIRS. And this is also looking at changes in blood oxygenation, but this time, instead of using sort of across your whole head, either X-ray or picking up on radioactive material or magnetic fields, this is just using infrared light. So they're using kind of like specialized infrared flashlights that they can shine on the surface of your skull, and they detect how that beam of light travels through your skull. Again, this is another thing that's a lot easier for me to describe when I can use my hands and show you my own head. But if you think of, so look at our image here. This is someone's brain from the top, so that's their nose there. So this would be the back of their head over here. And if you had your weird infrared flashlight, we'll say that's what that is, and you shine your beam of light onto the surface of the skull at just a little bit of an angle. If this beam of light passes through your skull and starts interacting with the fluids around your brain, it's going to bend that light so that it comes out on the other side at a slightly different angle. And this is a pretty extreme angle, but it gets the idea done. If we see a lot of bending, then we know that there's not a lot of oxygen in that blood. Whereas if there isn't as much bending, maybe it comes out like over here, then we'd say that it's very oxygenated. And so you would have your light shining device, your infrared light shining device, and then on the other side you have some kind of detector that would tell you what angle the light is coming out at. And they call it the attenuation of re-emerging light. And attenuation just means how much that beam's path was curved. So is it mostly straight or has it bent a little bit? This is far more extreme than what you'd see in real life. But by measuring how bendy the light beam has become, you can tell how much oxygen is in the blood in that part of the brain versus other parts of the brain. So by telling if there's oxygen there or not, it's going to give you an indirect measure of brain activity. So again, we're looking at changes in blood oxygenation, where if you're seeing a dramatic drop-off in the amount of oxygen in an area, means that that part of the brain is probably using that oxygen because it's active. So we can make some inferences about how active a certain part of the brain is, and then we can get these maps of which parts of the brain are higher activity versus lower activity. I'll clear that off. Our next different type of brain imaging is electroencephalography, or EEG. And this is one that we're pretty familiar with, but it's one that people tend to forget about in terms of brain imaging, because this is what it produces at the end. So it's not actually a picture of our brain in the traditional sense. This is actually telling us about electrical activity at different locations across our brains. So for an EEG, you're measuring electrical activity using electrodes on the scalp. So maybe you've seen within the department, there's a couple of people who do EEG work, and it looks like kind of a shower cap with a bunch of white circles and wires coming off of them. And each of those white circles is an electrode. And as we've talked about with our neurons, we know that neuronal firing involves a change in the electrical current. So we're seeing a difference in charge across those cell membranes. And these electrodes pick up on any changes in electrical charge underneath the part of the scalp that this electrode is on. They can be inter or intracranial, so most of the time we're going to see them outside of the skull. If you were, say, doing brain surgery and you wanted to test for activity in the area that you're working on, you could use one inside the skull, and it would be far more accurate, but it isn't feasible to cut into people's heads and put this inside their skull every time you want to do this measure. So you can do them outside as well, and that is the most common way to see it. But if you think of, so each line here, if we start at the very top, this line is one electrode, and you as the researcher would know where each electrode has been placed. So maybe this one electrode at the top has been placed on your right temple. And then the one below that would be placed a little bit above that. And the next one above that. And you know how they're mapped out. And maybe you'd place them in a spot that corresponds to an area of the brain you're interested in. Maybe over a particular lobe that you care about. Or maybe you're going to put them everywhere. You can have hundreds of them. And each will detect the changes in electrical activity directly below them. And unlike all of the other functional type brain imaging that I've talked about, where we have to wait for that blood to flow towards an area, where we have to wait for oxygen to get used up, where there's just a little bit of a delay, with EEG, we're measuring the neural impulses. We're measuring that change in electricity that's happening as soon as it happens. So our resolution for time on this is phenomenal. And you can see a change within milliseconds. Basically, as long as it takes for the signal to travel up the wire to your machine that's recording all of these squiggles. And for these squiggles, when they're, it's actually probably, again, easier to draw this. If they are very small squiggles, we'd say that there's very little activity. There isn't any neural impulses, there's no firing, there's no action potentials if they're pretty much flat. This would just be sort of background brain activity. But when we start seeing these big swinging things like in here, that would be telling us that there's lots of neurons firing in that area. And if we look on our EEG at the bottom here, you can actually see that we have really high responding here, but some other areas, like say this one line here, doesn't have lots of activity. So they might have asked a person a question at this time, and only certain parts of the brain are showing really high responding in response to that question. So we could then say that these parts of the brain are involved in us formulating a response to whatever question was asked. So that's a different kind of take on looking at brain activity. And for our last one, this one is even more out of left field. It's, again, not giving us a picture of the brain, and it's not doing what an EEG is doing, where it's telling us what's going on in a particular part of the brain. It's its own thing. But this one is my favorite, and this is transcranial magnetic stimulation, or TMS. And TMS is going to induce electrical activity using a magnetic field, which is a really fancy way for saying that they take a very strong, very small electromagnet and they can hold it up to parts of your skull, and it will interfere with your neurons firing because the neurons are firing using those electrical signals, and a strong enough magnet in just the right place can actually mess with that firing. So it can cause them to fire, it can keep them from firing. It's mostly just going to disrupt our cortical neural tissue. So it's going to disrupt functioning in the part of the brain that that magnet is applied to. And so if you're putting two and two together here, I am describing a situation where you have a participant come in, you can hold a magnet to part of their brain and effectively turn off the functioning in that part of their brain. And so one of the most interesting ways to see this happening is by deactivating part of the occipital lobe with the visual cortex. So it's responsible for how we see things and interpret the things that we're seeing. And so they would use, say, a letter detection task. So you'll have a participant come in and you give them a button. And on the screen, you're going to show them a bunch of symbols. And if they see a letter, they push the button. If they see a number, they don't push the button. If they see a punctuation mark, they don't push the button. So they only push the button if they see a letter, hence letter detection. And so you can look at their proportion of correct responses. So how often do they push the button when they see a letter? And what happens? And so here they have you pulse, or you apply the magnet before the stimulus is shown. And so this point right here would be sort of at the 60-second or the 60 millisecond mark. They would pulse the magnet, and then they would show you a letter, and you'd struggle a little bit. You'd still do kind of better than chance. You'd be sitting at like a 60% correct. You'd do okay, but not great. And then as soon as they turn the magnet off again, your performance goes back up to pretty much perfect. And then here, what they're doing is they're turning the magnet on after you've seen the letter on the screen, but before you can tell them that you've seen that letter. So you will visually see it, but then you can't do anything with that information to figure out that it's a letter. And in that case, you see this massive drop-off in performance, and they're down to like a 10% correct responding rate. And that's worse than chance. And the particulars of the study that they use to show this don't really matter. What we care about is that there's a type of imaging, quote unquote imaging, but there's a technique where you can deactivate different parts of the brain to see how your behavior, how your performance in different behavioral tasks changes when different parts of your brain are deactivated. Now, of course, this comes with a couple of caveats, things that a researcher using this technique would have to be aware of. That being, you don't want to go and deactivate part of the brain that's responsible for important bodily functions. So you'd want to stay away from the brainstem. So you want to make sure your magnet isn't so strong that it can get to your brainstem, because otherwise people might have trouble breathing. And also, I should note that these exposures are in sort of milliseconds, so there's just a fraction of a second where these parts of the brain are deactivated, and as soon as the magnet is removed, the neurons can go back to firing normally. So this isn't a long-term disruption, it's just a very, very quick thing, but it is a way for us to check which parts of the brain seem to function with which behaviors.